The paper written by Andrew Leslie and Kevin Boyce covers the ovule function and the evolution of the angiosperm reproductive innovations. Some quick background information. In 1827, it was noted that the primary difference between angiosperms and other seed plant groups involves the function of their ovules during reproduction. In the diagram to the right, it is labeled that the ovules are inside the ovaries and they're seed or egg-like. This paper tests two questions, the first one being, how do the functions of the ovules in seed plants constrain morphology, development, and resource allocation? And the second one being, how has the ovule structure evolved? The ovule function and structure. So all seed plant groups have similar structure. They have nucellus tissue, which produces and encloses the embryo sac, integument, which surrounds the nucellus with a small opening through which pollen grains will pass, and the gymnosperm ovules have a larger variety of functional roles during the reproductive cycle. Here's a diagram of the information that they collected. It pretty much said that the smaller the ovule, the less of a function the ovule had, and in contrast, the larger it is, the more elaborate the outer protective layer would be. In this diagram, the integument and nucellus are shown in black. The embryo sac is represented by the white ovule inside the structure, and the embryo itself is depicted in the black structure within the embryo sac. Here's another graphic showing a positive relationship between ovule width and ovule length, and it shows that pretty much over time the ovules have gotten slightly bigger, with the black dots being angiosperms, the open dots being living gymnosperm, and the triangles being fossils. Here's the ovule evolution. During the Carboniferous period, it possessed complex ovulate morphologies that appear to reflect a general tendency for seed protection and dispersal to be performed by extraovular structures and organs. During the Jurassic, it was nearly fully enclosed by a structure called the cupule, and it played a role in seed dispersal. And during the Mesozoic, extraovular tissues developed more robust with tightly packed seed cones. In conclusion, according to Leslie and Boyce, many of the innovations that characterize angiosperm reproductive biology have led to fundamental changes in how the ovule functions during reproduction. Organs such as the carpel have assumed roles in pollen capture, seed protection, and seed dispersal, performed more directly by ovules themselves and other seed plant groups. This study was done by Frankie D. Jackson and James G. Schmitt. It was done on vertebrate egg abnormalities from the Cretaceous period. The taxa being observed in the study were from the Cretaceous period, which lasted 79 to 80 million years. All points dated from here on are pulled directly from the paper. This includes both bullets on the PowerPoint and further in-depth explanations provided verbally. The four main taxa observed in the study were a turtle, hadrosaur, a sauropod, and a theropod. The first was an unidentified turtle taken from the Judith River Formation located in Hill County, Montana. The second was a hadrosaur that was taken from the Willow Creek anticline, which is in the Two Medicine Formation located in Teton County, Montana. The third was a sauropod taken from the Yucca Mahueva locality located in the Anasato Formation in the Neuquén Province, Argentina. The fourth was a theropod taken from the 7 Mile Hill, or the first fine microsite, which is located in the Two Medicine Formation in Teton County, Montana. The questions being asked are if there are any abnormalities in the eggs that were found by comparing them to other eggs found in the same locale, and if there are any pathological condition found in the eggs. They are also wanted to determine if stress was a factor in how the eggshells were formed. There were at least 13 eggs that were found in a weathered clutch in the Judith River Formation. Some of the eggs found were normal and contained embryonic remains, while others were abnormal. Figure A shows a normal turtle eggshell that contains embryonic remains. Figure B shows an abnormal turtle eggshell with two superimposed layers. The white triangles in the image show the upper surface of the inner eggshell layer, while the white arrow shows an elongated pore behind the impression. Figure C shows aragonite crystals inside the inner eggshell layer. The white triangle shows the contact between the inner and outer layer. There was only one hard-to-store egg collected, and it had identical microstructure to other hard-to-store eggs and eggshells. Figures A, B, and C are all the same egg found. Figure A shows a thin section of the eggshell in a plane polarized light. Figure B shows the same section of the eggshell, but under crossed polars. Figure C shows an enlargement of the spherulites at the outer shell. It shows that there is an absence of the calcite nuclei. There also appears to be a calcite-filled pore in the upper left of the photo. There are two eggshell fragments collected from two different egg beds. Figure A and B are from one egg bed, and C and D are from another. The black arrow in Figure A shows preserved calcite nucleation sites. Figure B shows the same eggshell but under crossed polars. The white triangles in Figure C show the shell unit nuclei. Jackson and Schmitt also point out that there is a lack of nucleation sites at the base of the outer eggshell layer. Figure D shows the inclusion of eggshell and permeabilization membrane between the first and second shell layers. The dotted white line outlines the membrane. This is a multi-layered eggshell that has two superimposed layers. Figure A shows a section of the two superimposed layers, which displays diagnetic alteration. Two thin external layers create the outer portion of the internal shell. The outer layer is missing where detachment of inner and outer shells occurred. Figure B shows the same eggshell just under crossed polars. Figure C is an enhancement of Figure A, showing the two superimposed layers and where the missing external layer is, which is at the top right-hand corner. Figure D is an enlargement of where the inner and outer layers meet. The white arrow points to the base of the lower layer. In extant taxa, adverse stimuli from the psychological or environmental stress can cause prolonged retention of her eggs, which in return can cause additional shell layers to be deposited over the eggs. This would create an unusually thick shell with superimposed layers separated by protein membrane. Multi-layer eggs are seen very frequently in turtles but are very rare in avian eggs. In extinct taxa, most of the egg abnormality fossils are actually fragments instead of in situ eggs or clutches. Multi-layered dinosaur eggs until recently were only found in 3 specimens, 2 eggs from the Upper Cretaceous and 1 from the Jurassic. In dinosaurs, the stress that resulted in egg retention most likely occurred immediately before the time of the normal oviposition. Studies suggest that the stress which produced egg retention by the female affected shell deposition during formation in the outer abnormal eggshell layer. In birds, stress of the sufficient magnitude may influence eggshell structure for a considerable length of time. In turtles, the physiological or environmental stress that produced the abnormalities most likely occurred before or during an early phase of the mineral deposition. The decreased shell thickness of an inner eggshell layer observed in the Cretaceous turtle egg is not typical of abnormal dinosaur eggs, nor has this morphology been documented in modern avian eggs. The reproductive anatomy is different between turtles, crocodiles, and birds. Turtles have 2 active ovaries and oviducts with simultaneous ovulation of ova. The uterus produces both membrane and eggshell. The oviductal wall next to each egg secretes the eggshell membrane first, then the aragonite mineral. Simultaneous shelling of several eggs occurs in the oviduct, and the entire clutch is laid at one time. Crocodilians show a divergence from other reptiles in the reproductive anatomy. The eggshell membrane is produced in one region of the oviduct, while the calcite eggshell is produced in a more distal area. The separation of oviduct function as the formation of eggs is shared with the birds and represents an archosaurian mode of reproduction. Crocodilians, however, retain the ancestral condition of the simultaneous ovulation of eggs and mass egg laying. Birds exhibit a sequential ovulation, with each egg shell and laid individually at a 24-hour or greater interval. These features are useful in determining if an eggshell was pathological. Multiple eggshell layers separated by permeabilization membrane, close confrontation of the base of the outer eggshell to the unweathered surface of the underlying egg, and structural relationships of eggshell features, which are things such as pore truncation, intercalated membrane and shell fragments, and optical continuity of the superimposed fragments. Definitive identification of any fossil egg, however, requires the presence of an in ovo embryo with diagnostic features. which makes classifying pathological eggs very difficult. A lot of the times, multi-layered eggshells typically result in embryonic death. The additional abnormal shell layers restrict embryonic gas exchange in the atmosphere and the embryo suffocates. Therefore, the multi-layered eggs that appear hatched produce uncertainty about the presence of a pathological condition in fossil material. While taxonomic identification can be a problem with the lack of embryonic remains, there are conditions to follow to make a reasonable taxonomic assignment. Stress produced on the female while carrying the eggs can cause the eggs to become abnormal. When making a taxonomic identification, it is important to keep locality in mind, as well as the inferences in physiology and anatomy and paleoenvironment of instinct taxa. It is important to remember that there are characteristics of biologic and taphonotic processes that may result in similar multi-layered eggshell morphology. This paper is titled Variable Growth Modes in Late Cretaceous Ammonoids, Implications for Diverse Early Life Histories. It studies fossil ammonoid invertebrates. Invertebrates are animals with no backbone. They are split into two groups, terrestrial and marine. Invertebrates make up about 90% of the world animal species, believed to have been around for over 3.5 million years. This study searches for patterns of septal angles within ammonoids throughout ontogeny. Fossil ammonoid shells were collected in Hokkaido, Japan, and 23 specimens belonging to 10 species of ammonoids. The earliest ammonites appeared during the Devonian, and the last species died out during the Cretaceous Paleogene extinction event. The studies looks at ammonoids from the Cretaceous period. Most ammonites have coiled shells. The chambered part of the shell is called a pharmacone. It contains a series of progressively layered chambers called camerae, which were divided by thin walls called septae. The last chamber is the body chamber. As the ammonite grows, it adds new and larger chambers to the open end of its shell. A thin living tube passes through the septa, extending from the body to the empty shell chambers. This allows the ammonite to empty water out of its shell chambers. Each specimen was polished along the median plane using silicon carbide powders. They were measured using a digital optical microscope. After they were measured, scientists determined that they would make two comparisons regarding the rotational angle and septal angle of the shell. First, the total rotational angle at each septum versus the septal number through ontogeny, and second, the septal angle between two adjacent septa versus the septal number through ontogeny. The late Cretaceous ammonoids examined in this study revealed that the ontogenetic patterns of septal angles are classified into four groups categorized by a single critical point in early, two critical points in early and or later ontogeny, cyclic fluctuations through ontogeny lacking a critical point, an almost constant septal angle throughout ontogeny and lacking a critical point. The critical point is a point where a new chamber has grown over time, or ontogeny. The various septal angle patterns in late Cretaceous ammonoids are in contrast with modern and fossil nautiloids, which have the common pattern displaying a single abrupt change in septal angles. Although the abrupt change of septal angles in nautiloids correspond with the hatching event from the egg, change of the septal angles in the examined ammonoids is hypothesized to correspond not to the hatching, but to the change from a planktic to a nectobenthic habit, demarcated by the post-embryonic stage. Therefore, the variable patterns of the septal angles within ammonoids suggest a diverse set of early life histories. The study is titled The Ontogeny of Two Species of Darwinuloidea and was conducted by Robin J. Schmitt and Takahiro Kamiya. Animals in this family have been present in the fossil record for over 350,000 years. There are approximately 30 species alive today in this family. These organisms can be found in many different habitats. These include wet places like rivers, ponds, lakes, groundwater, and even fallen leaves. The two species being used for this study are from the Darwinuloidea family. They are Darwinula stevensoni and Vestelanula. The development of an antenna-like appendage is what sets the two organisms apart from other members of the Darwinuloidea family. These organisms have been alive, have lives spanning from 1 to 4 years. Scientists are generally interested in the growth of these animals because they are one of the few asexual organisms that were recorded as long ago as the Carboniferous period. Questions have been raised recently because there have been a few very rare discoveries of these organisms to look to be male. The questions being tested in this paper are: what changes can be seen in the growth and development of two species that makes them different than most others in their family, and where in the growth process can they be recorded? Specimens used for the purpose of the study were taken from freshwater sources. They were studied and analyzed under an electron microscope. Darwinula stevensoni has 8 major stages of growth. In the beginning of its growth cycle, A1, it begins to develop reproductive organs within its body. In stage A8, the full appendage is developed and visible. Each appendage is generally has 4 to 5 segments, each moving differently. The fully developed antennae are covered with a carapace or a thick layer of skin similar to that of a turtle shell. Appendages are called walking legs and claws. In early stages of growth, because of the way that the antennae are positioned, it is unclear whether or not they are capable of full motion, but they are evidence that it may be able to create a current of water through its carapace, meaning that as water moves through its body, it can gain more of a range of motion. The addition of the other feathery appendages in later stages, there is better evidence for full motion. It can be concluded that these antennae are used for movement in most cases. It is unclear whether or not these antennae are strictly for movement or somehow are also linked to reproduction organs within the two species. There is evidence that the structure of the antenna is disrupted when the species lays eggs, giving more evidence that they could have less to do with reproduction than movement. In this video, we discuss growth and reproduction in plants, vertebrates, invertebrates, and microfossils.